in lecture on Tuesday. All right, but I want to just wrap up chapter 16 here. So with this information, then when we see it again on Tuesday, you'll definitely understand it a little bit better. Okay, so obviously we all know that the ear is responsible, all right, for our special sense of hearing, okay, audition they call it, and the cranial nerve that is responsible for this is your cranial nerve number 8, vestibular cochlear. All right, so that is going to be your primary neurons will run in the vestibular cochlear nerve. Okay, so we're going to talk about the different parts of the ear. So we're going to start with the most external portion, the external ear. Okay, made up of the auricle. We labeled it as the pinna, P-I-N-N-A. Okay. That's that big dish that sits, or funnel that sits on the outside of your ear. All right. Basically, it helps to funnel the sound waves into the ear canal, but it also acts to help protect the external auditory meatus too. Okay. So then that ear canal portion is the external acoustic or auditory meatus, or you can call it the external acoustic canal. All right. You can label it either or. But that is that passageway that leads all the way to your eardrum. But don't call it that on the test. All right, it is the tympanic membrane. All right, and in that ear canal, we have ceruminous glands. All right, and these glands are going to produce your earwax. Now, I know it says here it helps to impede microorganism growth, but if you let it get bad enough, when I say bad enough, if you let your earwax or your cerumen accumulate enough, you can actually cause quite an obstruction. And did I tell you the story about my roommate when we were living down in the Caribbean? I had a roommate. I was down there for about 3, almost 4 years, and he refused to clean his ears at all. All right, yeah. And his reasoning was because he wanted to let enough of his cerumen to collect because he was terrified of spiders. And he had heard on the island there that spiders crawl into your ear. So he said, well, they're not going to crawl in my ear. I'm not lying to you, for 4 years. I won't get into some of the details, but anyways, the man, yeah. Hear? Pardon?
Could he even hear?
Oh, yeah, believe it or not, he could. He could. Yeah.
Yeah, I don't think like you hear anything. They build up so fast.
Yeah.
It's insane. I can't.
Yeah, I've never tried. I know there's an ear treatment that you can use where you, I don't know, you use like a candle. Have you guys heard about that?
Yeah, my mom used to do that. It feels so good.
Yeah, I've heard good things about it, but I don't know. I've just been.
You're not supposed to do it like twice a year or something.
Right.
Yeah, but I heard it was kind of like mental. Like it doesn't actually do what it says it does.
Oh, really?
Yeah, it's kind of like placebo effect, I guess.
As long as it gets rid of some of it, because everyone says don't use Q-tips. You just impact it, you mash it in there. But anyways. And then our last structure is the eardrum, the tympanic membrane. It is made of epithelium, though. But its job, it's much like a drum or a tambourine. All right, when the sound waves strike it, all right, it will start to vibrate. But more importantly, it starts to help to amplify sound waves. All right, so when we're in lecture, we'll talk a little bit more about that. Okay. All right. Oh, yeah, that's right. These are out of order. Give me a second. Bam, bam, bam. There we go. All right. So these are the 3 parts to the ear. The external ear we'll talk about, the middle ear, and then the inner ear. So you can see here's our external ear, all right, with the external acoustic meatus all the way down to the tympanic membrane here. And then in the middle ear, not too many, but we have the ossicles there, okay, the malleus, the incus, and the stapes. And then we also have the eustachian tube, okay? And then don't forget, we've got the tensor tympani muscle here, okay? A couple other structures, but that's what we're going to talk about, all right, for the middle ear. And then when we get to the inner ear, you can see all the different structures. We've got, all right, our Boney Labyrinth, all right, which is pretty much, the Boney Labyrinth is all this white structure or ivory structure. Okay, it's got the cochlea, and then it's got the vestibular complex here. But it includes all this whole thing, all right, is going to be the Boney Labyrinth. And we'll break these down, okay? And then you can see cranial nerve 8, all right, right here, all right, which is the the vestibular cochlear nerve, it enters in. But look what's close to it, facial nerve. Okay, facial nerve comes right in, okay, with the vestibular cochlear nerve, because they're very close to each other also on the brainstem, all right? And I'm sure you've been studying that, and you probably noticed the same thing. But the facial nerve, all right, is going to travel in very close approximation with the vestibular cochlear nerve. There's a tumor that you can get which is called an acoustic neuroma, and it's a tumor that usually grows right around the vestibular cochlear nerve, and it can cause, obviously, balance issues, hearing issues, but it can also cause facial tics, depending on how big it is, because of the facial nerve is close by. Because remember, facial nerve is the motor component for all the muscles of facial expression. Okay? All right, so we'll hit all those different parts. I'll refer back and forth to that slide. All right, our middle ear, okay? This is that space in between the external and the inner ear here, all right? And that space we refer to as the tympanic cavity here, all right? So remember these two structures, the oval window and the round window, all right? On the internal portion of the middle ear, you've got this portion of bone there, that bony wall there, okay? And in that bony wall, all right, you have these two structures, all right? And both of these structures have to do with the cochlear duct, all right? And we'll talk about that in a little bit. All right, so we have the auditory tube, also known as the eustachian tube, all right? And that's that tube I was telling you about, all right, that leads down to the nasal pharynx. So if you have, and one of the issues with that connection, all right, one, its primary job is to help equalize pressure in your middle ear there, all right, especially when you're flying in a plane, or driving in a car, and you happen to go up the side of a mountain, okay? We use it to help to equalize the pressure, but one of the problems is too, since it connects to the throat, if you have a throat infection, like strep throat, for example, all right, those microorganisms, that bacteria can crawl up, all right, or move, I shouldn't say crawl, but it can move up the eustachian tube and it can cause an ear infection, all right? So that's one of the things that you have to be careful of, all right? And one of the ways that you can help to clear your ears, all right, when you're when you want to equalize the pressure, yawn, chew gum. Someone told me chewing gum when you take a flight is a good way to help keep it from your ears popping. A lollipop? How many licks does it take to get to the center of a Tootsie Roll Pop?
I don't know.
Yeah,
You got-
the patient.
You've never seen the commercial?
Yeah.
Oh, okay.
With
Yeah.
Al?
Yeah, now, I think he says 3. He's like, he licks it 3 times and he bites into it. All right. So our 3 auditory ossicles, all right, the malleus, the incus, and the stapes. The malleus is going to be the one that sits just on the other side of your tympanic membrane. Now, these are the old names that they gave them. They used to call the malleus the hammer, and the incus used to be called the anvil, and the stapes used to be called the stirrup, all right? And it's listed in your lab notebook as that too. Don't use those, all right? Use these, okay? All right, then the middle auditory ossicle is the incus, and then the stapes, to me it looks like a staple, but this is the structure that attaches, all right? The base of it's got this foot plate here, right? It's an oval-shaped foot plate, and that oval-shaped foot plate is going to fit nicely into the oval window, because as the bones start to vibrate from sound waves, they're going to transmit that sound, and it's going to pass through the oval window, and on the other side of the oval window is a fluid-filled cavity. So sound travels best through a solid, and then it travels second best through liquid, and then air is the worst media for it to travel through. It can't travel through vacuum, all right? And that's why there's no sound in space. They can't hear you scream in space. All right, so their purpose is to amplify those sound waves, all right? And they do so through vibration. And like I said, as the vibrations, all right, are moving back and forth, it will then transmit that vibration energy into, all right, the fluid on the other side of the oval window, okay? Now, it's kind of cool that your inner ear, or excuse me, your middle ear has 2 muscles that are in place to help protect it from excessively loud sounds. You got the tensor tympani, and this is spelled wrong, by the way. It should be an I there. All right, you've got the tensor tympani, all right, and then you've got the stapedius muscle. And again, they both help to restrict the auditory ossicles from vibrating too much if a loud sound goes off, okay? Because that can get really amplified and that can cause some damage. All right, so here you can see them all. All right, here's the tympanic membrane. All right, here's the malleus. It's connected right to the tympanic membrane. Then you got the incus here, and then you got the stapes. And then right here is the oval window. So as all of these are vibrating back and forth, all right, it's going to create these pressure waves, which will then be transmitted into the cochlea, all right, which we'll talk about here momentarily. That's the middle ear for you. All right, inner ear. The inner ear is located in the petrous part or portion of the temporal bone. You remember that structure? All right, on the inside there of your skull, okay? And the inner ear, right, externally, we see what we call the bony labyrinth and the membranous labyrinth, all right? Now, hold on one second. I want to go back and show you. There it is. Okay. All right. So this again is the bony labyrinth. All of this yellowish ivory colored structure. All right. That's the bony labyrinth. Okay. So, and then just there's a certain part, you can't see it from this, but on the inside of the bony labyrinth, that's the membranous labyrinth. All right. So the bony labyrinth, think of it like the labyrinth is like a maze. Okay. Lots of little rooms and compartments. And that's what the bony labyrinth is, all right? And in these little compartments and spaces there, it's filled with this perilymph, all right? Very similar to interstitial fluid, okay? It's very similar to that. Then, in one of those compartments inside of this bony labyrinth, we have what's called the membranous labyrinth, all right? It is a membrane-lined, fluid-filled tube inside. I'll show you a nice cross-section of it. Okay, and inside of that, we've got what's called endolymph. It's very similar to the intracellular fluid inside a cell. So that means it's going to have high amounts of potassium. Okay, very similar to intracellular fluid. All right. So this endolymph is very important when we talk about hearing and equilibrium. We'll talk about that. All right, because when those sound waves vibrate the auditory ossicles, they'll start the stapes will start to beat against the oval window there, and those, like how you would bang against the tambourine, and that creates pressure waves. And on the other side is this endolymph, and so those pressure waves get transmitted through the endolymph. Like if you're sitting in your tub, you're starting to make waves in the tub, all right? Or if you've ever been in an above ground pool and you all want to make a whirlpool. Has anyone ever done that? When you walk around the outside of the pool, you know, and it starts to, then you start to swim against the current. Same type of concept, all right? It's going to create these pressure waves, all right? And that plays an important role, especially when we start talking about sensory components of this. Okay, so in the inner ear, we see three structures or regions, all right? The first is the cochlea. Okay, that's going to have to do with hearing, and then the other two, the vestibule, okay, which is made up of both the utricle and the saccule. They're two sac-like structures, all right, and they have to do with your head position, okay, like looking down, looking up, and looking straight ahead. And then you've got your semicircular canals, those ring-like structures, okay. So the important structure that we want to keep in mind for the cochlea, all right, is going to be the cochlear duct, all right. And that's going to have that endolymph in there that we're going to talk about. So now what we're going to do is we're going to do a transverse cut through. Remember what I said? Remember the cochlea was that snail shell structure? All right. And basically what it is, it's a long tube-like structure, okay, that has three smaller subcompartments inside. And what we've done is we've just rolled it up to make it look like a snail shell. Okay, So what this drawing is showing you, for example, here's our cochlea. It's all rolled up. We're going to unroll it, okay? So we're going to unroll the cochlea, and then I'm just going to take a transverse section out of it, like that. And that's what this is. Okay? And so if I zoom in, you can see that there's 3 compartments here. Okay? You got your scala tympani, all right, your scala vestibuli, all right, and then you've got your cochlear duct. And we'll hit all these compartments again. All right, we're going to talk mainly about that right there, all right, our cochlear duct. And that's inside, all right, of the membranous labyrinth. All this other stuff is in the bony labyrinth, because this is the bone here. So this is going to be perilymph, perilymph, this is endolymph. Okay? All right, and then you can see how we have a nerve here, all right, that's coming from this structure here. Remember the spiral organ? That's the actual hearing organ, all right, and then we'll talk about how that generates the actual sensation of hearing. Okay, and then over here, this is all the vestibular apparatus over here. So we've got our semicircular ducts, and then we've got our vestibule, all right? So what we've done is we've just taken a cross section here of one of our semicircular ducts, and it's the same type of thing. You got all these little compartments in here, okay? If you see it's blue, that's going to be made up of the perilymph fluid, and then this one compartment here, that's going to have the endolymph. And we'll talk about that when we cover the vestibular system. All right, so let's jump into the cochlea. We're going to stay with hearing for right now. Okay? So there's a couple structures that we didn't label. All right? We labeled the scala vestibuli, the scala tympani, all right, and then the cochlear duct. All right, that is right here. All right, there's the cochlear duct. This is the scala vestibuli. All right, and this is the scala tympani. Sorry, brain fart. Okay, so a couple structures that we didn't see before. All right, the modilius, all right, which is what we refer to as the bony axis of the spiral. So we have our, I can't draw very well. All right, here is your snail shell. All right, and the bony axis just goes right through the middle of it. Okay, that's where the nerve enters, and then you have your nerve branches that come off. Actually, let me just show you. I got a picture here. What am I doing? All right. All right. So here you can see the modilius entering in, all right, and then it gives off these spiral ganglionic nerve fibers that go to different parts, all right, of our cochlea. Okay, so you see all these little branches here. Okay, so in the cochlear duct, the floor of the duct is going to be the basilar membrane, and the roof is going to be the vestibular membrane. We labeled those last time in lab, right? So that's these structures right here. Okay, here's your basilar membrane. Basilar is easy to remember. It's on the floor at the bottom. Okay, and then your spiral organ sits right on top of that basilar membrane. And then right above the spiral organ, you have the tectorial membrane. And that's what those hair cells were attached to. All right, and then you've got your cochlear duct right here. And then the roof of the cochlear duct is the vestibular membrane. Okay, so you get your scala vestibuli and then your scala tympani down here. Okay, that's it for the ear. Just showing you the structures. I'm not going to be getting into the physiology really, all right? This is the lab portion, so we're just going to focus on the anatomy. Okay? All right, let's finish up and talk about the last of our special senses, the equilibrium here, okay? So keep in mind, all right, equilibrium and balance are a little bit different than one another, okay? So when we talk about equilibrium, we're really talking about our awareness of our head position, of where it is. If you're looking down, if you're looking up, if you're looking straight ahead, if you're looking side to side, all right? Equilibrium, all right, has to do with that awareness of those positions. It also has to do with balance. Now, here's the thing. your vestibular system, balance and equilibrium, okay, it gets a lot of help. It gets a lot of help from your visual system, all right, your eyes, because that gives you a lot of input of where you are, all right, and it also gets help from our proprioceptors. Remember all those receptors around our joints and tendons and muscles? They're constantly giving your brain feedback as to where your body is. But if I sit here completely motionless, all right, and I close my eyes, what I've done is I've taken my visual system out of play here, all right, and then if I'm not moving my skeletal muscles or my joints or my tendons or anything, my proprioceptors aren't involved, okay? So I know that I'm looking straight ahead because of my vestibular apparatus is telling me that, okay? So now I have my eyes closed and obviously I know what I'm doing. But if I'm looking down here, okay, I know I'm looking down because actually I moved and proprioception was giving me that feedback as I was looking down. But once I stop moving and I'm in this position now, I know I'm still looking down because my equilibrium, my vestibular system is providing my brain with that information. All right, and we're going to talk about how it does that, okay? So yes, it helps, all right, with balance, all right, but it's not the main function, all right? It's not the main system that provides that balance, okay? So our vestibule is going to help, or the utricle and the saccule, they're going to help with static and linear acceleration. So what does that mean? All right, static is when I'm looking straight ahead. My head's looking straight. Okay? And then with linear acceleration, it's when I look down and I look up. That's it. Okay? So those two structures are going to provide my brain with that information of static equilibrium and then linear acceleration, up and down. Okay? The semicircular ducts, these guys here, okay, these guys here, all right, they're going to deal, all right, with, where are we? Whoops, wrong one. With like rotation, what we call angular acceleration, okay? Twisting my head from side to side, okay? They're going to help deal with that, okay? And you have three of those structures there, those semicircular ducts, all right? You've got this one that's up and down, all right? That's the anterior semicircular duct. And then the one that's kind of horizontal, obviously we call that the horizontal semicircular duct. And the one that's kind of like an oblique angle, that's the posterior semicircular duct. Okay? So when we talk about head positions of static equilibrium and linear equilibrium acceleration, we're going to be focusing on this structure right here, the macula. That is going to be our receptor organ, all right, that is located, all right, in the saccule and the utricle. All right, both of those together, as you recall, are called the vestibule here. Okay, so inside of this macula, you have these specialized hair cells. Okay, and it's a nice layer. It's almost like a carpeting. All right, and then obviously in between these specialized hair cells, you'll have your supporting cells. Okay, but on these hair cells, and I'm not even going to try to draw this for you. I'm just going to show you the picture. All right, on these hair cells, all right, you have this structure that's called a stereocilia, okay, and it's a modified cilia, okay. Then you have this one special one that's called a kinocilium, all right. So all these cilia, all right, they project out of the cell, okay, and what they do is they sit in this glob, all right, that's called the otolithic membrane. And it's like, it's not as thick as gelatin, okay? But it's almost like a gelatin, okay? And it sits in there. And then you have these structures that float. Well, why am I drawing it? I'll just show it to you. My drawings are awful. Sometimes I try to impress myself, but it never works. All right? All right. So this is what I'm talking about, all right? So here's the macula, okay? So the macula is made up of these hair cells and then the supporting cells. And then obviously you've got these sensory neurons here, okay? So on top of the macula, you've got this big glob of gelatin there, all right? That's called the otolithic membrane. And inside, you have these structures or these things called otoliths, and they're just crystals. So if anyone here has ever talked to somebody that's ever had benign positional vertigo, does anyone know what that is? It's vertigo, you know, you get dizzy, you know, and you have a lot of difficulty trying to maintain your balance. All right. But benign positional vertigo, most of the time, the causes are these things, these otoliths. You get an accumulation of them, and they just bang into these receptor cells, and that puts your balance off. All right. And they just keep colliding because there's a huge amount, and they get stuck there. And you can't get them out unless there's two different types of maneuvers that you can do with the patient, in which you can move their neck and head and their body. All right, it takes about 10 minutes to go through a series of maneuvers, and you can move those little crystals out of there so it should stop doing it. It's kind of neat. So you can see here on the hair cell, you've got this structure here. All right, that's called the stereocilia. I'm gonna move over here for you. Okay. So you can see it's kind of like a tapered structure, right? So all these little ones here, all right, those are the stereocilia, and then at one end, you got one individual stereocilia with this big bulb on it, and that's called the kinocilium, okay? More on that in a second. You guys pretty good with that then? All right, here's your hair cell, all right? It's got the cilia here, and one cilia, the one at the one end, has this big structure up there called the kinocilium. Okay, so in that gelatinous layer, all right, the otolithic membrane, all right, we have those calcium carbonate crystals, the otoliths. So what happens is your head moves as it tilts, all right, if you look down or if you look up, all right, think of it like being on the deck of a ship. Things start to sway, all right, and if you've ever seen a show or a movie in which stuff wasn't latched down to the deck of the ship, when the ship sways to one side, everything slides to that side, and vice versa. When it sways the other way, everything slides. Well, that's what's going on in this otolithic membrane there, all right? So as you're moving your head, all right, that gelatin, all right, will move forward, and those crystals will move forward, and they'll bang into those receptors and stimulate the receptors. I'll show you. There you go. So, all right, so here you are in your normal upright position, looking straight ahead. Okay, so the otolithic membrane is just hanging out here. Okay, so when it's just in this position, okay, the nerve cell here is going to set off nerve signals at a regular interval. Okay, so they're always just going. But when it's at this regular interval here, this standard nerve frequency, your brain knows that you're looking straight ahead. Okay, so let's just say it gets 10 action potentials per second. Then it knows, okay, 10 action potentials per second, the head is looking straight ahead, okay? So now, all right, what I do is I'm going to look up towards the ceiling. And as I look up towards the ceiling, all right, then all that gelatin and those otoliths move down like that boat, all right, and it sways down. And everything shifts towards the kinocilium here. And when it does, all right, it releases more neurotransmitter. So instead of getting 10 action potentials in a second, it gets 60, let's just say. It increases, all right? And when that happens, then your brain knows, okay, the head is looking up, okay? Now, to flip it, say you look down now, everything swings in the opposite direction, like that boat tilting to the other side. All right, everything moves away from that kinocilium, all right, and that hyperpolarizes, all right, the nerve cell. So that means it will release neurotransmitter a lot less, all right, it decreases it, or it doesn't release it at all, okay, and now the action potentials of the nerve signals that get sent to the brain are maybe 2 a second. So your brain knows, okay, they're looking down. Okay, so there's always a nerve signal being sent, all right? So when it's the normal frequency, all right, like it's shown here, then your brain knows, okay, I'm looking straight ahead. When it increases a lot, like we see here, then your brain knows, oh, I'm looking up. And if it slows down or is nothing, then it knows you're looking down, okay? So there's always some neurotransmitter being released, all right? But it just depends on how many signals are occurring. We'll talk more about that in lecture. All right, let's finish up with the last part, that's angular acceleration. Okay? What happens when you rotate your head from side to side? Okay? So we're going to deal with another type of receptor organ, and that's going to be called the ampulla. Okay? Remember on those semicircular canals here? There we go. We can see a little bit better. All right, on our semicircular canals, you can see these swellings here. Those are the ampulla. Okay, you do have to know that for lab, excuse me. Okay, so they're at the base of each of those semicircular canals, and they have got a structure inside, all right, called the crista ampullaris. And I think that structure looked, well, I was going to try to draw it for you. It looks like a flame to me, like a blue flame. All right, here's your crystal ampullaris, and it's a gelatin type of structure. Okay, surrounded here by endolymph. Okay, and then at the base of it, all right, again, you have your sensory hair cells with the same type of scenario. All right, they've got the stereocilla and the kinocellum. All right, in that same configuration. Here's that kinocellum, and then you have all these other stereocilla, excuse me, and they just go down in size at a decreasing interval. Okay, and it sits on those cells again. Okay, with the nerve coming off. Okay, same type of scenario, except this time you have a whole bunch of them, all right, and they're surrounded by this big gelatin structure there, all right, and that's referred to, all right, as the crista ampullaris, all the hair cells and the support cells, and then it has that gelatinous copula there, all right, I call it the blue flame, okay? So what happens is, when you rotate your head, when you turn your head from side to side, that endolymph, that fluid, all right, starts to bang into the blue flame, all right, into that crista ampullaris. And as it moves it into a specific area, all right, so if it bangs all those stereocilla toward the kinocellum, then we get depolarization. And if the stereocilla moves away from the kinocellum, we get hyperpolarization. Regardless, okay, same type of scenario, neurotransmitter will be released, and depending on whatever that frequency is going to send that information to your brain to tell you if you're looking to the left or you're looking to the right. That's basically what we're looking at, all right? And the cranial nerve, all right, that operates this or receives that sensory information is the vestibular cochlear nerve, cranial nerve number 8, all right? But this comes from the vestibular branch, all right? We labeled that last time. Okay, we'll talk more about this in lecture. So last part here, just a quick pathway here for equilibrium. Okay, those sensory signals that are generated, all right, from both the macula and or the crista ampullaris are going to travel down the vestibular branch of cranial nerve number 8. All right, that's going to be the primary neurons, and they're going to head to two places. All right, it can either go to the vestibular nuclei, right, in the brainstem, or it can go to the cerebellum. Because remember, cerebellum has to do with proprioception, balance, fine motor movements. Okay? So if it goes to the vestibular nuclei and the medulla oblongata, all right, it's going to actually play a role in reflexive eye movements and also some of the balance there. But also our cerebellum, because some of the information goes to the cerebellum, that's going to help with balance. Not only that, but also with some of the muscle tone to help maintain balance. Because if you're trying to correct your balance, you're going to contract certain skeletal muscles while relaxing others. All right? And then from those structures, all right, it's going to travel up to the thalamus, all right, and then past the thalamus, I think we all know where it's going to go, up to the cerebral cortex. Okay? Same type of scenario. Again, we'll talk about this a little bit more in lecture, but here you can see the vestibular complex, all right, cranial nerve 8. Is going to send some information here to the medulla oblongata. Some's going to go to the cerebellum. Then the cerebellum is going to send sensory feedback information back down to the skeletal muscles. All right, deal with proprioception to help with balance. All right, while others then we're getting input from, all right, the eyes here, because remember your visual apparatus is going to help with balance and equilibrium. All right, and that's all going to play a role here in the brainstem. And then from the vestibular nuclei and from the cerebellum, all right, the secondary neurons are going to transmit information up to the thalamus and then eventually up to the cerebral cortex. All right, we'll get into more detail. I just kind of wanted to give you a little bit of an introduction. All right, kind of get you excited over the weekend for what you're in store for on Tuesday. Tuesday, yes. All right, so Tuesday, we're going to finish up this chapter and lecture, and then we'll do some review in lab. Please, if you have some questions, because we can do some lab and lecture. I'll do some test questions for you for lab, and then if there's any questions on lecture stuff, you can ask me in the lab portion. Okay, but try to think of